Hello my dear students, I welcome you to NEET World. So in this session, we will be starting the acids, bases and salts. So this is from 10th class CBSE, right? Okay. So now we are very much familiar with acids, bases and salts, and we are using them in our daily life also. So it is very important to know about these acids, bases and salts. So let us start our session without any late. Okay. So before starting the session, just click on the like button so that it will be really boosting us to do more videos on this type of topics. Okay. So let's start our topic with a like button first. Okay. So here, what exactly acids? So this acid, the word acid is a Latin word, so which means acidus, which means sour to taste. So generally acids are the compounds which are sour in taste. Okay. And these acidic substances converts blue litmus to red litmus. So what exactly litmus, we will study about that. It's a litmus paper, we will be studying about that. So what happens? Acids are the compounds which converts blue litmus to red. Okay, and these are sour in taste. Okay, so whereas bases, so these are bitter in taste. So what is the taste of bases? So they will be having bitter in taste. Okay, and the next, so these bases converts, so red color litmus to blue color. So red litmus converts into blue color. Okay, so these are the basic terms you can remember. We need to study more about these acids and bases in the upcoming sessions. We will be discussing more deeply. So let us understand the basic terms first. Okay, so let me take a color pen here. Yes. So now look here. So what are the acids we are using in our daily life? For example, lemon juice we will be taking. So what is the nature of that particular compound? So it is having citric acid. What it is having? So lemon will be having citric acid. That is what the citrus fruits we are saying now. Okay. So the next, curd will be having lactic acid, right? The next, vinegar, acetic acid. Okay, and the next, see here, unripened fruits like grapes, here we have, and tomatoes. So all these are what? Acidic in nature. What they are? Acidic in nature. Okay, and the next, on the other hand, we have washing soda. So what is the formula of washing soda? Na2CO3. So which is basic in nature. What is the nature of washing soda? It is basic. Okay, lime water. So what is this lime water? So these are also basic in nature. Like so they will be having, soapy touch and bitter taste, soapy touch. Okay, so these are the two characters you can remember for bases. Okay, so I hope it is clear, like you got some little basic idea. Let us see more things about these acids and bases. Okay, so now in order to identify, see, I will be giving a compound to you. Okay, so I have given a solution. So I just telling you to identify whether it is acid or base. So how do we identify this? Okay, so to identify this, we have something called indicator. So indicator are generally compounds which will use it to distinguish, or which will use it to identify whether a given compound is acid or base. Okay, so whenever we add indicator to that particular solution, so there might be some color change. So depending upon the color change or the characteristic smell, we can able to identify whether this is an acid or the base. Okay, like that we will be using the indicators. Let us study about that very clearly. So indicator is a chemical compound which is added to the solution in very small amount to detect whether it is acid or basic in nature. The given solution, whether it is acid or base, how can we detect? By adding the small amount of this indicator. So what do we mean by indicator? It is just a compound. It is just a compound. Okay, I will tell you, I will show the structure of the indicators also. Just hold for some time. So by the end of the session, you will get complete clarity on these indicators, acid, bases and all. Okay, so the next, acid base indicator is a substance which possesses one color in acidic medium and the different color in the alkaline medium. See, alkaline medium, or we can also call it as basic medium. Okay, both are same here. Okay, so indicator is what? It is a substance which will be having different color in different mediums. For example, it might be colorless in acidic medium, but it will be colored in the basic medium, like this. Now what happens if the solution is acidic? There will not be any color. if the solution is basic, whenever we add this indicator, the indicator will be having some, that will be showing some color change or the color in that particular solution. So by looking at the colors of that particular solution, when we add the indicator, we can able to identify whether it is basic solution or acidic solution. Okay. So indicators are basically colored organic substances either extracted from plants. Okay. So when we are extracting this indicator from plants, we can say them as natural indicators, or synthesized in the laboratory, so we can call them as synthetic indicators. Okay. So I will show you what are the natural and what are the synthetic indicator examples also in the upcoming slides. Okay. We will discuss everything one by one. Just look here. So now in the beginning of the class I told you that there is something called a litmus. Okay. So litmus paper I told you. So what exactly litmus? So it is most commonly it is used as natural indicator to detect acids and base. Okay. So this is a natural indicator used to detect whether it is an acid or base. Okay. So this litmus is of two types. How many times I am saying? It is of two types. Okay. So one is blue litmus, another one is red litmus. Okay. So when I am saying litmus, so simple litmus, it is a neutral solution. Okay. If I say litmus solution, it will be neutral solution and it has purple color. What is the color of neutral litmus solution? It will be purple color. Okay. And acid converts this turns blue litmus to red. So whenever we have a blue litmus paper and when we are putting a drop of acid onto the litmus paper, or take this litmus paper and dip into the acidic medium, this blue litmus dip into the acidic medium or acidic solution, what is the color change you will be observing? This blue litmus converts into red. Okay. So this is what happens here. Okay. Similarly, when an alkali turns when an alkali turns red litmus to blue, if you keep one red color litmus paper into the basic medium or alkaline medium, so that converts into blue color. So what is this colors indicating? The change in color says the respect to nature. If a blue litmus converts into red, I can say this is acidic solution. Okay. And if a red litmus converts into blue, we can say it is basic solution. That's it. Okay. I hope it is also clear up to here, right? Now acidic substances turning blue litmus solution to red. So what are the solutions? Vinegar. So vinegar contains acetic acid now, so that converts blue to red. Okay. So now lemon juice and orange juice converts blue litmus paper to red litmus paper, or blue litmus solution to red color solution, right? Then next, juice of unripened mangoes, tamarind, that is what imli we are calling, right? So that converts, that is also having tartaric acid. What is the acid? Tartaric acid converts blue to red. Hope it is clear, right? Then basic substances turning red litmus solution to blue. Okay. So basic substances turning red litmus solution to blue. So what are the solution? Baking soda solution, washing soda solution, and next bitter gourd, next cucumber extract. So these are having basic in nature. Therefore, whenever we dip the litmus paper into these solutions, so what they will do? They will convert red litmus to blue litmus. Red color to blue color, right? Hope it is clear now. Okay. So let us look into the indicators that we have. Okay, here. So these are the things we have seen about the litmus paper. Now we have told that, that there are some indicators now. So indicators of two types. Indicators are natural indicators and one is synthetic indicator. So let us see some natural indicators. Here we have two things. One is red cabbage juice. So red cabbage juice means it is purple in color in neutral medium, okay, and turns red or pink in the acidic medium. Okay. So when this purple color in this red cabbage juice is having purple color in the neutral medium, okay, and this and turns red or pink in the acidic medium. So purple color changes to red or pink in the acidic medium. In basic medium or alkaline medium, its color changes to green. So purple color changes to green when the solution is basic in nature. For example, I have a beaker in which basic alkaline solution or basic medium is there. This solution is basic. Now what I am doing, I am taking this red cabbage juice a little bit and I adding to this solution. Now what happens? since this is basic in basic nature, so whenever we are adding this purple color neutral cabbage juice, so what happens? Purple color changes to green color, so it indicates the solution is basic in nature. Hope it is clear now. Yes. Next, turmeric juice. So turmeric juice is yellow in color and remains same both in acidic as well as neutral medium. Okay, there is no color change when we are keeping that in the acidic medium or when we are keeping it in the neutral medium. In the basic medium, what happens? Its color becomes reddish or deep brown. Okay, so yellow changes to red color in the basic medium. Okay, so this is a just a normal like activity you can do at your home also. Just take some turmeric powder, so keep in the, like just put it in the water and make it as a solution. So now what you will do is take some amount of detergent powder so that you can add it. So detergent powder generally having some basic nature, right, or any basic compound you can take, just add that. So you will be seeing red color. Okay, so during some events, occasional events like something like marriages, you will be seeing to the legs, they will be applying turmeric powder and along with some surf detergent powder. So you will be seeing some red color on to their legs. So that is what the color change you will be observing. I hope it is clear, right? Some other indicators here, these are synthetic indicators. One is phenolphthalein, another one is methyl orange. Okay, so what exactly phenolphthalein? So this phenolphthalein actually it's an organic compound. This is also an organic compound. Okay, so now they will be having very big structure, I will show you, and this is generally it's a weak acid. Phenolphthalein itself is a weak organic acid. Okay, so now let us study this. In neutral or acidic medium, okay, it is colorless. So what is the color of this phenolphthalein neutral or acidic medium? It is colorless. Okay, while in basic medium, the solution, the color of the indicator changes to pink. So see here. So let us say I have a beaker in which acidic solution is there. So to that, if I add this phenolphthalein, is there any color change? No color change. It will remains as such. Okay. Now in another beaker, I have basic solution. So to that I am adding phenolphthalein. So you will be seeing that colorless solution is colorless indicator is converting into pink color. So this is what the color you will be seeing in the basic medium. In acidic medium it will remains colorless. I hope it is clear without any confusions, right? Hope everything going well, right? So now I will show you the structure of phenolphthalein, just hold for some time. Next we have methyl orange. So this is weak organic base it is. Okay. Okay, methyl orange is colorless, orange colored dye. Okay, it is orange colored dye and keeps this color in the neutral medium. Okay, so it will remains as orange color in the neutral medium, and in acidic medium the color of the indicator becomes red. Acidic medium it becomes red and in basic medium becomes yellow. So this is what the color changes you have to remember. So acidic medium becomes red, in basic medium it is become yellow. Which one it is? Methyl orange. Okay, in neutral medium orange color, acidic medium red, basic medium yellow. You can remember this. Okay, so this is a phenolphthalein structure. If you see this, see these are the phenol groups we can say. Okay, these are the hydrogen, this is the hydrogen, these are the two hydrogen which are responsible for their acidic nature. Okay, so whenever this H plus ion comes out, okay, so this structure will become like this. Since I am saying it is acidic, so it releases H plus ion. Okay, we will study why it is releasing H plus ion, because acidic, like definition of acid itself, release of H plus ion, we will study about them in the upcoming classes. Just listen here. So this is acidic compound which we are saying it as phenolphthalein. This is the structure, original structure. It is colorless in its acidic medium, like this it will be. Okay, whenever it releases this H plus ion from here, it will become like this. Okay, that H plus ion is removed here, right? So now this is called quinol form. What is that? Quinol. This is called quinoid form. Okay, so in the acidic medium it is colorless, but when it comes to basic medium it will be showing pink color. This is the color change. This is the color change you will be seeing when a phenolphthalein is added in the basic medium. Hope it is clear, right? Okay, so you need to remember this. So there is a JEE question also. What is the structure of phenolphthalein in the acidic medium and in the basic medium. So in acidic medium you should call it as quinol form, in basic medium you should say it as quinoid. So it is quinoid form, here it is quinol form you can say. Okay, these are the words you have to remember. Then next, so we have another type here, olfactory indicators. What is this olfactory indicators? Indicators giving different odors in acidic and basic medium are called olfactory indicators. So this is based on the odors. Okay, indicators which are giving different odors in acidic medium as well as basic medium. So we are calling them as olfactory indicators. Okay, some natural olfactory indicators are this one. Like see, onion ordered cloth strips. So what you will be doing is you just take some onions, like a piece of onion. So just chop it into the small pieces. Now keep that in the that onion, chopped onion with some cloth strips. Okay, in a cover, and place it in the fridge for 12 hours. Okay, so now what happens after 12 hours? That cloth strips will be smelling like onions, right? So this is what the this is what the onion ordered cloth strips. You have prepared onion ordered cloth strips. Now the onions have a characteristic ammonical smell. Okay, what is the smell? Ammonical smell you can say for onions. Okay, which is retained in the acidic medium. So now that cloth strips, now dip it in the acidic medium. You take any acidic solution and dip it in that. For example, HCl you can take, hydrochloric acid, dip that onion strips. So what? The smell will remain the same. Okay, but if you dip the same onion cloth strip in the basic medium, the smell of that onion will be lost there. So what is the difference here? The smell remains same in the acidic medium, but smell of that onion strip cloth will be lost in the basic medium. So this is what the order, difference in order we are seeing now. Okay, so different in orders in acidic and basic medium are called olfactory. So this onion ordered cloth strips can be considered as one of the example for olfactory indicators. And another one is vanilla essence. Okay, another one is clove oil. Clove oil also one of the olfactory indicator. So vanilla has characteristic sweet smell which is retained in acidic medium, but it is destroyed in basic medium. Yes. Okay. So in acidic medium remains same, but in basic medium it will be loses its sweet smell. Okay. So I have few questions to you. So you need to say me this. Okay. We already discussed. So based on that, these are exercise questions you can consider. Okay. What you will observe when methyl orange is added to dilute hydrochloric acid. So what I told you, methyl orange is a weak acid now. So when it is weak acid, base now, methyl orange is weak base I told you. So when we are adding this dilute like HCl, like dilute hydrochloric acid, what happens? So methyl orange color converts into red color in acidic medium, and orange color converts into yellow color in basic medium. This is what the color change you will be seeing. So in orange, what happens? Orange color converts into red color in acidic medium. That's it. A drop of phenolphthalein is added to the solution of lime water. So tell me, lime water is basic in nature and phenolphthalein is acidic compound. So what happens when phenolphthalein is added to this basic solution? This solution will be now appears in the pink color, right? I have shown you that colorless one test tube and a pink color solution. That is what happens in this case, in the second case. Hope it is clear, right? Now a knife which is used to cut the fruit which was immediately dipped into the water containing drops of blue litmus solution. So knife is dipped into the blue litmus solution. Okay, the color of the solution changes to red. Yes, blue litmus solution converts into red color. So what does it is indicating? The knife which we use it to cut that fruit, might be fruits are having acidic nature, that's why now acid is like attached to this knife. Therefore when we are keeping in the blue litmus solution, the knife is now appear to be red in color. The solution becomes, what is the solution becoming red. So what is the nature of the fruit? Yes, what is the nature of fruit? Yes, I already told you, blue converting into red means what is that? Acidic. The fruit is acidic in nature. That's it. Very simple, right? Hope everything is going well. Yes. So what is the name given to the indicators given giving different orders in acidic as well as basic medium. Yes, so this is your homework question for the day. Okay, so this is your homework question for the day. So I will be waiting for your answer in the comment box. So what is the name given to the indicators giving different orders in acidic as well as basic medium? Yes, I will be expecting answer from you for this question because we discussed this very clearly, right? Yes, I hope you will get you got the answer. Just write in the comment box so that I will respond to your comment in the chat box. Okay, thank you for watching. Keep learning. Happy. Have a nice day.